All right. Well, I think that that is enough time for people to join, but maybe some people will come later. So I guess we'll get everybody to mute themselves. And thanks so much for coming. I feel I should preface this to begin with that this is in no way sponsored by AHS. So this is purely sponsored by Noel. I really wanted to give this talk initially because I thought we would be included in getting the vaccine at some point, and I wanted to know more about it and what was in it. And I have Nick and Rye at my disposal to ask my questions to, but not everybody has a microbiologist in their life. And as it rolled out quite quickly and sort of I had some of my questions answered, I realized people, friends and family were coming to me as a nurse asking me questions, and I didn't have a good enough understanding in order to talk about this in an accessible way to those people. So the two aims I'm hoping from this we get out of it is having more understanding of the vaccine that we're receiving, and maybe having some ways that we can engage the public in explaining what this vaccine is and why it's so important. So as introductions, this is Nick and Rye, and they're PhD candidates from the Shriver Lab at the University of Calgary. Nick is developing a vaccine against Haemophilus influenzae, and Rye is developing vaccines against respiratory pathogens in pigs. Other credentials include Nick is my boyfriend and Rye is a very good friend of ours. So to cover what we'll talk about today, we're going to just talk quickly about the COVID virus and vaccines in general, and then specifically mRNA vaccines and how they work, the Canada Health requirements for a vaccine and trial requirements, and then specifically the Pfizer vaccine, as that's what we're getting. And then we'll cover the questions that you guys sent me. We'll keep all of those questions confidential. As we were just mentioning, it might go a little over time, but like there's no attendance list. If you need to go somewhere or don't want to listen anymore, you can just leave. And if you want to hang out after, I think Nick and Rye are keen to answer questions. So if you think of something, you can write it down or write it in the chat or send a direct message to me, and we'll cover the questions at the end. Okay, I'll let these guys.
I think just important to know, it is being recorded right now. Is that okay with everyone? Or?
Oh yeah, good point, Rye. I suppose please get off the call if you're not okay with it. There were some people at work who were hoping to listen in, so hence the recording.
Okay, I guess I'll share my screen here. Okay, so as Noel said, my name's Nick, and I'm a PhD candidate at the University of Calgary in the Department of Microbiology, Immunology, and Infectious Disease. So before I start, I just wanted to establish some common ground. So first of all, it's normal to have concerns surrounding vaccines. So, and actually, scientists share this sense of caution, and it's the reason we do these large-scale, thorough clinical trials to assess safety and efficacy. So it's ultimately the vaccine we're administering to the public eventually is safe and effective. And secondly, we all want good health outcomes for ourselves as well as our friends and family. And vaccines are actually the most effective medical intervention we have to achieve this goal, and thus put an end to the current pandemic, which is something I'm sure we all want. And lastly, I just wanted to say that I don't have any conflicts of interest. I don't benefit financially from giving this talk. And I work on vaccines as part of my PhD, but I don't receive any funding from the companies developing any of the COVID vaccines. So I wanted to just show what vaccines can do. So these are three of the nastiest illnesses we've ever seen. So measles, polio, and smallpox. And through vaccination, we've actually been able to eradicate smallpox completely, almost eradicate polio, and significantly reduce the annual incidence of measles cases. And you can see here the impact that the introduction of the first measles vaccines had, and recommendations, the impact of recommendations for dosing had on the reduction in the number of cases over time here in Canada. And you can see here that I wanted to show sort of the dangers of vaccine hesitancy. So in 2011, we actually had 752 cases of measles here in Canada. And it just shows you that anytime there's complacency with regard to preventing these illnesses that predominantly affect young children, it's very easy for a resurgence to occur in some of these infectious diseases when there's a reduction in uptake of these vaccines. Nowadays, though, in the last couple of years, it's gone down to about 25 cases per year, and people are now starting to talk about eradication of measles again. And this is the same graph, just without the measles data. So as I said, we eradicated smallpox. The WHO, smallpox was eradicated in North America in 1952, and eventually eradicated worldwide in 1980. And you can see the impact that the inactivated polio vaccine, which was the first widely available vaccine, the impact that had in the mid 50s on number of cases of polio, and as well as the availability of the second polio vaccine that's still widely used today, the oral polio vaccine, the impact that's had in sort of drastically reducing the cases of polio, which is, of course, a devastating disease that can cause paralysis, and one that we don't even really see anymore because of vaccination. So next, I just wanted to introduce coronaviruses in general. So just so we're on the same page in terms of terminology, COVID-19 is the disease, and it stands for coronavirus disease 2019 because it was first identified in 2019. And the virus causing the disease is SARS-CoV-2, and that stands for Severe Acute Respiratory Syndrome Coronavirus 2. And SARS-CoV-2 is a member of a family of viruses known as the coronaviruses, and the scientific name for the family is Coronaviridae. And these are a group of RNA viruses that cause disease in mammals and birds. RNA viruses just referring to the fact that their genetic material is encoded by RNA as opposed to DNA, which is the case for all other living organisms. And I'll explain that further later on in the talk. Some examples of coronaviruses that infect humans are SARS-CoV-1 or SARS coronavirus, which I'm sure we all remember. It caused outbreaks of severe respiratory illness in 2002 and 2003. It originated in bats, was transmitted through intermediate hosts, namely civets and raccoon dogs, which are small mammals that are found in China. And the case fatality rate for SARS was about 10%. Another coronavirus that causes disease in humans is the Middle East Respiratory Syndrome coronavirus, also known as MERS-CoV. And this first emerged in 2012, and sporadic outbreaks have occurred since then. It also originated in bats, but is transmitted through camels. And it has a much higher case fatality rate, around 34%. So it's a very severe illness. SARS-CoV-2, of course, is the virus that's causing the current pandemic. It originated in bats as well. However, the intermediate host is unknown, and whether there is an intermediate host is unknown. And the case fatality rate is actually much lower than the two other coronaviruses I mentioned. It's around 1%. It's estimated to be around 1% when you take into account all of the asymptomatic cases and cases with milder symptoms. And ironically, that's actually been the low case fatality rate has been the main reason that this pandemic has sort of exploded into the global phenomenon that we've seen. And it's because people are walking around with mild symptoms or no symptoms at all, not knowing that they have the virus, and therefore spreading it to other people unknowingly. Whereas with these severe diseases, they're much easier to contain because people have to go to the ICU, and it's much easier to quarantine people in a hospital room, as I'm sure you're all aware. And lastly, I just wanted to mention that there are a few common cold coronaviruses which don't cause serious illness in humans, and they've been circulating for decades throughout the globe, and they cause an illness similar to the common cold. And lastly, I just wanted to draw a comparison to the influenza virus, or the flu. So the flu has been sort of adopted as a term to describe, as a colloquial term to describe several different ailments, but really the correct way to use it is to refer specifically to the infection by the influenza virus, which is also an RNA virus, but it belongs to a completely different family called the Orthomyxoviridae. So so COVID-19 obviously is quite literally not the flu. It causes a very similar disease as SARS-CoV-2, but it's a completely different virus in a different family. And it actually has even some of the pandemic strain, the so-called pandemic strains with pandemic potential, their case fatality rate is still only around 0.1%. And the seasonal flu is much, much lower than even 0.1%. So COVID-19 is despite being less severe than these other two coronaviruses, it's still a much more virulent pathogen than the seasonal flu. And just yesterday, Dina Hinshaw confirmed as much by announcing that COVID-19 has now killed more Albertans than the last 10 flu seasons combined. So now I just want to introduce the concept of RNA by talking about what's called the central dogma of molecular biology. So what this refers to is the fact that the genetic material of all living organisms is encoded in DNA, a molecule called DNA. And this encodes all of the instructions needed to make the proteins, which are the molecules that actually carry out the cellular functions that we need to keep us alive. And there's actually an intermediate step where DNA is converted into RNA, which is which is sort of a single-stranded iteration of DNA that allows proteins to ultimately be made. So it's sort of an intermediate intermediate set of instructions that's used in the process of making the proteins we need to carry out our cells functions. So DNA is converted to RNA through a process called transcription, and that occurs in the nucleus, which is this sort of core compartment of our cells that contains our DNA and sort of protects it, contains and protects it. You can think of it as sort of the house of our cells versus the cytoplasm where translation occurs, which is the process of converting RNA to proteins, is sort of the backyard of our cells, or the surrounding property of our cells. So DNA is converted to RNA in the nucleus through transcription. The RNA is then shuttled out to the cytoplasm, where molecules called ribosomes convert the RNA into proteins. And another analogy for this is sort of to think of DNA as a cookbook, RNA as a specific recipe in that cookbook. So the cookbook is a book of instructions, a book of all the instructions you'll need to make whatever final meals you'd like. And then the RNA is a sort of specific set of instructions, and then proteins are the final product. So the final meal resulting from following the instructions written in the cookbook and the recipes. And so how does RNA relate to SARS-CoV-2? Well, instead of using DNA to encode its genetic material, its genetic set of instructions to make it all of its proteins, it encodes its genetic instructions in an RNA molecule. So it sort of skips a step. The term plus strand RNA just refers to the fact that it's all ready to be used to make proteins through translation. And another way of saying that is by referring to this RNA molecule that's all ready to be translated as messenger RNA or mRNA. So you can think of plus strand RNA as synonymous with mRNA. And so these sort of protrusions on the surface of the virus are called spike protein. And the instructions to make these spike proteins are encoded in the genetic material, which again is RNA. And this RNA, these instructions for making the spike protein, as well as the spike protein itself, like the protein, not the RNA, have been used, are the targets of all of the vaccines that are under development for COVID-19. So apologies for the busy slide, but I just wanted to highlight sort of what happens when the virus SARS-CoV-2 infects our cells. So what happens first is the virus uses its spike proteins to bind to a molecule on the surface of human cells called angiotensin converting enzyme 2. So the spike protein binds to ACE2, and that's what allows it to get into the cells. So it sort of buds into the cells in these sort of bubbles called endosomes. It eventually uncoats and unpacks its genetic material, and that's released into the cytoplasm, at which point the fact that it's positive strand RNA allows it to be translated right away, allows the virus to hijack our ribosomes to make its own proteins. What happens then is it makes its proteins, makes copies of its RNA, and then packages all of those molecules into new virus particles. Those virus particles then exit the cell and go on to infect other cells. So the sole raison d'etre, so to speak, for a virus is to make more copies of itself and to infect other cells. So it basically comes into our cells solely to make copies of itself by hijacking our ribosomes and our translation machinery.
Nick, is this just happening on any cells that have ACE2 receptors?
Yes. Okay, so next I want to go through the types of vaccines. So I've divided it up into the more traditional vaccines that we've seen sort of pre-COVID, as well as the non-traditional vaccines that we're seeing with the advent of some of the SARS-CoV-2 vaccines in the past year. So the first of the traditional is a live attenuated vaccine. So an example of this is the measles, mumps, rubella vaccine, as well as the oral polio vaccine. And this is just basically a weakened form of the virus or bacterium. So it's alive. The pathogen is alive and able to replicate, but it's sort of weakened in a way that it doesn't make us sick, but still induces an immune response that will give us protection against the disease. Another traditional vaccine platform is an inactivated but whole killed vaccine. So this is the entire virus, but it's been killed in some way such that it can't replicate and can't get us sick when it's administered to us. So an example of this is the intramuscularly administered inactivated polio vaccine. And lastly, there's the subunit or recombinant protein vaccines, which are just a piece of the virus or bacterium, as opposed to the whole thing, as was the case in these first two platforms. So usually it's a recombinant protein, such as the surface protein on hepatitis B, or some of the surface antigens on the that are used to target some of the meningococcal strains, meningococcal serotypes that cause meningitis in people. And so moving on to the non-traditional, so the first is the viral vector strategy, which is used by the Oxford AstraZeneca vaccine, ChAdOx. And so what this refers to is the fact that a viral vector, which is a virus that can't infect humans. So in this case, it's the chimpanzee adenovirus. That virus that can infect humans is used as a delivery system for the antigen, for the genetic material encoding the antigen, which in this case is the spike protein. Another non-traditional platform is the DNA vaccine platform. So basically, the DNA encoding the instructions to make spike proteins is injected as part of administering the vaccine. And there are actually 4 SARS-CoV-2 DNA vaccines in development that are either in phase 1 or 2. And lastly, which is the main focus of this talk, it's the mRNA vaccine, which is an mRNA molecule that encodes the instructions to make the spike protein. And as you know, the Pfizer, BioNTech, and as well as the Moderna vaccines both use this technology. So I showed you what happens when SARS-CoV-2 infects your cells, but this is what happens when you're injected with the mRNA vaccine. So the mRNA vaccine actually comes packaged in these pockets of fat molecules, these vaccine lipid droplets. So that's what's actually being injected into your deltoid as part of the IM route of administration. So the lipid droplets are injected into your muscle. It's taken up by cells that are surrounding the injection site. In most cases, it's immune cells such as dendritic cells and monocytes. So focusing on dendritic cells, those cells take up the droplets. The droplets sort of bud into the cells, similarly to the way the SARS-CoV-2 gets into the cell. It buds into the cell, the mRNA gets released into the cytoplasm, and as a result of the fact that it's mRNA, it's in the mRNA form, it's all ready to be translated. So our ribosomes can immediately get to work translating the instructions encoded in the mRNA into spike proteins. And so what happens next is once these spike proteins are made, they're then displayed on the surface of the cell by molecules called MHC, class 1 and class 2 molecules. And what these do is they serve as a sort of alarm system to notify the sort of commander of the immune system, the helper T cell, that there's a foreign invader. So dendritic cells, which is the cell that's for the most part taking up these viral vaccine lipid droplets, the dendritic cell serves as a sort of security guard that through the display of antigens on its surface serves as a sort of sentinel for the immune system, alerting it as to the presence of foreign invaders. And so that's what it's doing here. It's displaying the foreign spike protein on its surface such that helper T cells can see it and be aware that there's a foreign invader. The helper T cells then alert these other immune cells called B cells, which are sort of the soldiers of the immune response of the immune system. And B cells are responsible for displaying and producing high affinity antibodies that are specific to whatever antigen the helper T cell tells the B cell is a threat. So it makes a bunch of antibodies that are specific to the spike protein on SARS-CoV-2. And the other sort of soldier of the immune system is another type of T cell called the killer T cell or cytotoxic T cell, which then, whose job it is to look for, be on the alert for human cells that are infected with SARS-CoV-2 as a result of the fact that the helper T cell has alerted the killer T cell about the threat that this virus poses. So it goes around, looks for cells that are infected with SARS-CoV-2 and destroys them if they find any infected cells. And the last important point that I want to stress here is the fact that DNA is protected in the nucleus of your cells, where the mRNA vaccine cannot interact with it. So, as I said, DNA is protected in the sort of house of your cells. It's locked up in the nucleus, whereas the translation of the mRNA, the instructions encoded in the mRNA in the vaccine, the conversion of that mRNA into the spike protein, that all occurs in the cytoplasm, or the surrounding property or backyard of the cell. And so there's no possible way for the mRNA vaccine to modify your DNA or your genetic information in any way, because it can't access the nucleus.
Sorry, does the nucleus just have a different, is the envelope made out of something different than the whole cell?
It's a similar constituency. It's just that there are these little pores that in the nucleus. So it's the same molecules that are making up the membrane, the outer membrane, outer plasma membrane, as it is the nuclear membrane. It's just sort of a subcompartment of the cell that has these pores that are sort of selective, so it only lets certain things into the nucleus. And actually, some other viruses are able to get into the nucleus. They've evolved ways to get into the nucleus, and in some cases change sort of insert themselves into our DNA. But SARS-CoV-2 doesn't do that, nor do any vaccines made to target SARS-CoV-2 do that either. Okay, so this is just to say that we've been investigating some of these non-traditional platforms for many years now. So you can see RNA vaccines have been explored for certain strains of flu that have been identified as potential pandemic threats, as well as the Zika virus, which I'm sure you remember caused defects in neonatal children a few years ago, mainly in Brazil. So an RNA vaccine was explored. RNA vaccines were explored for that virus, and some of these other platforms, such as the viral vectors and DNA vaccines, have been explored for several other viruses, such as Ebola and HIV. Namely, so most of these clinical trials were for pandemic response purposes. So in case there was ever a large outbreak or pandemic for caused by any of these viruses, they were sort of at the ready to be deployed if any of these viruses were able to cause a pandemic. And so these are some of the examples of clinical trials looking at mRNA vaccines. So again, Zika virus, an mRNA vaccine targeting Zika virus was explored. Some mRNA vaccines targeting some pandemic potential flu strains were explored. And then this other virus called chikungunya, a vaccine for that was also looked at. And for this is an old paper, so most of these have been completed already, but it looks like mRNA vaccines as a whole are highly safe and in most cases immunogenic. And the recent trials for the Pfizer and Moderna vaccines, which Rai will talk about later, they've confirmed this. Like these mRNA vaccines are highly safe and highly effective ways of preventing disease by pandemic potential viruses. And this is just referring to the clinical trials that Moderna is doing. So Moderna's whole shtick is basically coming up with mRNA vaccines. So as you all know, the mRNA vaccine for COVID-19 is basically ready to go in most places, or almost ready to go in most places. Cytomegalovirus is another respiratory pathogen, and there's a mRNA vaccine in phase 2 targeting that virus. And again, there's a Zika mRNA vaccine targeting Zika in phase 1 of clinical trials. So the past couple slides were just to say that we've been investigating the possibility of using mRNA vaccines for a while now. And despite the fact that sort of this is the first time they've been administered to the public, that doesn't mean that we don't know anything about them. We've actually, mRNA vaccines has been established as a viable platform for many, or identified as a viable platform for many years now. And just to show some of the advantages of RNA vaccines, so it's a versatile platform. It's easy to scale up production for producing these vaccines in large amounts. It induces strong immune responses. It's easy to produce, as I said. Delivery of a minimal vaccine construct is possible, so that just refers to that the only thing you're injecting is the target antigen, or the instructions that encode the target antigen. You can administer it more than once, it's safe, and it's immunogenic. And this has been this is an old paper, as I said, so the immunogenicity can be updated to very strong, as we've seen in the clinical trials that have occurred this year. And then lastly, I just wanted to sort of compare the side effects that that are popping up in the news, or will be popping up in the news likely once more and more people get the Pfizer vaccine. And you can see that it compares pretty favorably to other vaccines on the market, such as the flu and Shingrix, and you can see even some people getting the placebo, which is saline only. They experience some of the same side effects as people getting the actual vaccine. So overall, it's a pretty comparable side effect profile to some of the other vaccines on the market. And that's all I had for you today. So I'll just pass it over to Rai.
Hello. Awesome. So I actually just learned that Moderna did get emergency usage authorization just like 30 minutes or an hour ago by the FDA. So we'll see when Canada does the same. So let me share my screen. All right, so my name is Rai, and I'm in the same lab as Nick, and so I'm also doing my PhD in vaccine development, and as Noel said, I look at some bacterial pathogens that cause disease in pigs. But today I'll be talking to you about sort of the COVID vaccine that some of you have already received, it sounds like, which is amazing. I'm very jealous. So let's discuss this Pfizer vaccine. So some of this information might be a little bit repetitive, but I think it's always good to understand the basics. And so here I've got the schematic of COVID, and what you can see when you look at COVID, like Nick was saying, is that COVID is an RNA virus. And so the genome of the entire virus is encoded in just RNA. And COVID, or SARS-CoV-2, sorry, when you think about it as a virus, it's kind of like a little ball. I had a plushie that is like COVID, or SARS-CoV-2, but it's in the lab, so I don't have it with me. But when you look at it from the outside, all you really see are these spike proteins, which is why they make such a great immune or a vaccine target, because that is something that the immune system can see and use that to actually recognize when the virus is in the body. Now, when you look at the entire RNA, so one of the questions I get is that, well, you know, this is a vaccine, it's an mRNA vaccine. Could getting this vaccine lead to us making and getting infected with the virus? And the answer is no, because if you look at the spike protein, that is only a small segment of the entire RNA genome that is inside this virus. And the vaccine only encodes or only has that small portion of this RNA segment. So this RNA segment that encodes the spike protein is essentially contained in these little fatty bubbles. And what that does is that it protects the mRNA, because mRNA is very, very unstable. And the other thing it does is that it allows the virus to actually enter our cells. So our cells have this fatty layer as well. And this fatty layer around the mRNA allows it to fuse with the cell and allow this mRNA to enter our cell. Now, as Nick was saying, the DNA is usually locked up in this nucleus. And so that's kind of that cookbook analogy that he was talking about. So you can assume that it's locked up in a cupboard. And then you've got specific recipes that you can write down on a piece of paper or a flashcard, and then you can bring it out into this kitchen area where you have all the ingredients in your pantry to make that dinner. So the mRNA, again, is that flashcard with the recipe for making the spike protein. And so when it's in this cell area or the kitchen area, the cell is able to actually make the spike protein with the ingredients it has inside the cell. So when that happens, the body is very, very good at recognizing when something is our body and when something is not our body. And so when the spike protein is made, our cells kind of go, hold up, this is not part of who we are. So what is this thing? This is weird. So let's just figure out a way to eliminate it, because we don't want to deal with anything foreign or anything crazy in case that's a problem. So essentially the cell treats the spike protein as foreign, the body treat the entire spike protein as foreign, and then prepare an immune response to it. So that is exactly what is happening to anybody who's gotten the spike protein or the Pfizer vaccine recently. And also mRNA, like I said, is very stable. It's not going to last in the cytosol for very long. It'll like last maybe 10 hours or less than a day. And then the spike proteins also won't last very long either. So that's what's happening to you when you get the vaccine. So what is actually inside this vaccine? So the medicinal ingredient is that mRNA that we were talking about that encodes the spike protein. And then there's a whole bunch of non-medicinal ingredients that sound very complicated, but essentially you've got a bunch of fats that make up that fatty layer. You've got a bunch of salts that help maintain the pH. So I know that when I was in the hospital once, I had gotten a whole bunch of saline IVs, and that saline IV is usually at a certain pH to make sure that it's physiologically similar. And so these salts kind of help do the same thing, maintain that pH. You've got sucrose, which is just sugar, and that's just keeping the fatty bubbles from sticking with each other. And then the water that goes in to dilute the vaccine. So the vaccine is supplied as something that's frozen. So you've probably heard that there's this cold chain requirement for the Pfizer vaccine. So this vaccine needs to be stored in -80 to -60 degrees Celsius, which is very, very cold and is not something that conventional freezers can do. So you need very specific freezers for this. So each vial has 5 dosages. And then what you do is you take that vial out, you thaw it, and then you add 1.8 mils of sterile saline. And then that vial will have 5 dosages of 300 microliters or 0.3 mil each. And then that could be stored at 2 to 25 degrees and has to be used up within 6 hours. So that obviously has some implications in where these vaccines can be delivered and who gets these vaccines. So the government of Canada and Health Canada have already decided that this vaccine is not going to be most appropriate for the territories up north just because of that cold chain requirement. So they might potentially be getting the Moderna vaccine instead. So I just want to shift a little bit over into vaccine development and approval. So we talked a little bit about what vaccines are, and I always try to explain vaccines as sort of like biological drugs that act as military drills to prepare your body for an invasion or an infection. And, Nick was talking about all these different types of vaccines, and there are different types of vaccines and different routes that you can immunize people, different adjuvants you can use, and they essentially are like different types of military drills, like the Navy or the Air Force or land-related Army stuff. So when vaccines are developed and people are looking at it for approval, there are really two things that people are looking at. And the first is always, is it safe? Like, that's the most important thing. And then you look at whether it works or not. And oftentimes it's even hard to know if a vaccine is working before it gets approved. And the reason for that is because a lot of times these infectious diseases are not that prevalent in the community. So if you think about something like meningitis, we don't have an outbreak of meningitis going on, but there are meningitis vaccines that are constantly being developed, and you have clinical trials testing these meningitis vaccines. Well, you can test that it's safe, but there isn't enough community transmission happening to make sure that it's actually effective, that vaccinated people in that small trial are protected compared to the people who've just gotten the placebo. So oftentimes what happens with effectiveness for a vaccine is that it is looked at after it's already been licensed and it's been given to the community, and then over several years after, we often figure out whether it's effective or not. But there is some preclinical data that goes into suggesting that it is effective, so they don't just like throw something in there and hope for the best. Like, there is data to suggest that it will work. And so when I say, what do we look for? Who is this we? So typically it's like the limited scientists that are working on that vaccine, typically, again, the regulators that are approving it, who are also people with a science background. In the case of the COVID vaccine, it's kind of literally just everybody. So you've got scientists and physicians who are working on the vaccine, but you also have scientists and physicians that are not working on the vaccine. The scrutiny on this vaccine is insane. And then you obviously have regulators at different levels of that development process who, again, also have that background in infectious diseases, immunology, etc. So again, that scrutiny, everyone is looking at this vaccine and looking at that data. And so this is kind of a bit of a schematic going into like what goes from start to finish in terms of developing a vaccine. So the first thing you want to do is identify that target. So in the case of the COVID vaccine, it's that spike protein or that spike mRNA. And so once that is identified, you want to do some preclinical trials, whether, so vaccines are working in animals and lab tests, some basic stuff like that. What does that target do? You do a lot of that research around that pathogen, that virus. Now, typically that R&D phase and that preclinical phase can take a very, very, very long time because you only have a couple of people that are working on this in the entire world. What was incredibly impressive was that the sequence of COVID or SARS-CoV-2 came out in January, and we had identified the spike protein and figured out so much about that spike protein in a matter of months, which is unheard of. And so that's why we were able to, as a global, group, start clinical trials, phase 1 clinical trials, as early as April and May. And so that's why we've been able to get there that quickly. So the clinical trials, there's usually 3/4. So there's phase 1, phase 2, phase 3. The phase 1 is just a small trial, and you're just looking at whether it's safe or not. So you're giving it to a small group of people, you're just monitoring for safety. And then phase 2 is, again, a bigger group of people, and you're monitoring for safety. And you might be also looking at whether it's a bit effective in terms of, are the people producing antibodies? Is there some of those immune markers that we would expect for people that are getting the vaccine? And then the phase 3 is usually a much, much larger study where, again, you're looking at safety as well as that effectiveness. And the reason why you kind of go from small to large is, again, you want to make sure this is safe in people before you move on to larger trials. And that larger trial really allows you to identify safety concerns that are rare. And so once you've completed all of this, that's when you apply to the regulators for a license or approval. And then after you've gotten that approval, there is also bodies and regulators in place to monitor that vaccine administration and the effects of that vaccine administration over years, essentially. And so currently where Pfizer, Moderna, and AstraZeneca are they're applying to regulators for that license. So Pfizer has already gotten the EUA in the States, they're approved in the UK, and they've gotten full approval in Health Canada. Moderna just got the approval in the States, and I think Health Canada is still deciding or discussing the results. And I think AstraZeneca is still also in discussion. But yes, it's quite impressive that, you know, we've gone from identifying that sequence in January to having 2 vaccines approved now at this point, at least in the States, in less than a year. And so how was the Health Canada vaccine specifically approved? And so usually what happens is that you put all of that data together, that phase 1, phase 2, phase 3, everything together, and then you apply for approval. But in this case, what Health Canada is allowing applicators to do is put forth their application on a rolling submission basis. So AstraZeneca and Moderna and Pfizer, they all actually submitted their applications back in early October. And what they've been doing is they've been updating Health Canada as results have been coming in. And when Health Canada had enough results that they thought it was okay, they approved it. And so Health Canada conducted an independent review of the clinical trial data. So they have essentially assigned a group of people, like a team per application. And that team includes, again, experts in toxicology, infectious diseases, immunology, microbiology, epidemiology, a lot of experts. And they comb through that data, and then they essentially recommend whether it should be approved or not. And with this independent review, they have agreed that the Pfizer safety and efficacy analysis is what they also agreed with. So what is that Pfizer clinical data that we've been hearing about? So this is kind of like an overview of the Pfizer clinical studies. And so initially they did a phase 1 trial, which again, you can see with very small numbers, 24 people with the vaccine and 6 people with the placebo. And that is, again, just looking at whether the vaccine is safe. And then you've got a much larger phase 2, phase 3 trial over here, where you've got in each group over 20,000 people recruited. And the type of trial is a randomized observer blind trial. So essentially, the trial participants have no idea if they're getting the vaccine or the placebo. The administrators have no idea if they're giving the placebo or the vaccine. So everything is blinded. And then that blinding is sort of lifted at the very end after they have already gotten all the results, just to make sure that there's no bias going into affecting that result. And what was also really interesting about the study is that how quickly they recruited 21,000 or 40,000 people. That is insane. It takes so long to recruit people for clinical trials. And then they also tried to make sure that they had a lot of people that are in some COVID risk categories. So 40% of their recruiting or their trial participants were over 56 years old, which is, again, part of that group of people that are more susceptible to severe cases of COVID. They also took people with chronic health conditions like hypertension, diabetes, asthma, because, again, those are groups of people that are more susceptible to severe types or severe reactions from COVID. And so that seemed like a really well-designed study. And they also tried to hit different demographics, so different race and ethnicity, gender, etc. What they did not include were women who were pregnant. They did not include severely immunocompromised people, and they did not include children under the age of 12, mainly under the age of 16, but they had very few people between 12 to 16. So that's why there's not a lot of data on that age group. But as they keep rolling out the vaccine, I think some of that data will come in. So what are essentially the results of this trial? And so you might have heard that the vaccine efficacy is 95%. And so this is essentially a graph showing that people are getting this first dosage here and they're getting the second dosage 21 days apart. And then you're essentially tracking people who are getting symptomatic COVID. And you can see that right here, the rates are pretty much the same in terms of people getting COVID till about 10 days after the first vaccine. That's when you see the vaccinated group start to taper off a little bit. And then after the second dose, again, like you don't see very much COVID appearing in that group. Whereas the placebo group continue to keep getting COVID, essentially. And so this is what the numbers are. So for symptomatic COVID, so mild symptomatic COVID, in the vaccine group, there were 8 people who got it. Whereas in the placebo group, there was 162 people who've gotten COVID. And then in severe COVID, you've got 1 in the vaccinated group and then 9 in the placebo group. So using these numbers, Pfizer was able to calculate the efficacy of protecting against COVID as 95%. And they're going to essentially continue following this group to see whether they what the protection looks like longer term. And so that number might change a little bit. But at the moment, it's very, very promising because regulators were really hoping for something that provided 50% efficacy protection. And so something that's close to 95, and even the Moderna one is around that number, is quite amazing. And so there is a bit of a safety follow-up. So right now, what we know is that Pfizer has looked at their trial participants for 2 months after the second dosage, and they essentially have plans to keep monitoring those people for up to 6 months post-dosage, as well as 2 years post the last dosage. So that's essentially to just see if there's any safety things that pop up. Right now, it doesn't look like there's no indication that will be the case, but that is again something that again happens with all vaccines, where after it's approved, you keep monitoring it to see what those effects are, or if there are any. And so, as Nick was saying, that there are some side effects that we can see with the vaccine. Some of them you might have already experienced if you've gotten the vaccine. So some reaction in the injection site, headaches, chills, fever, muscle pain, tiredness, and joint pain, a lot of which were also seen to some extent in that placebo group. And then one of the things that this is like a news article. The schematic is really nice, but the reason why I wanted to show this was because they mentioned that severe reactions were reported in up to 4.8% of individuals, and it was likely after the second dosage. And I thought that was kind of interesting because the FDA document, there's just a 59-page document that Pfizer submitted that the FDA used to approve it. In that document, it only says 0.5%. So I'm not sure where this 4.8% is coming from in this article, and I do not know if they just have a different terminology that they're using, or if they just mean these specific symptoms just felt worse after the second dosage. So I think that's just something that we all have to be cautious of, I think, going forward, is that there's always going to be perhaps not necessarily intentional, just unintentional misinformation, or just something that's not as clear as it could be. So the vaccine is in Canada is approved for 16-year-olds and older at the moment. I didn't see anything about, when I was looking at Health Canada's website, anything about people not being allowed to get a vaccine, but they did have some warnings and precautions. So if anybody has any allergies to those ingredients that I was showing for the Pfizer vaccine, or a history of serious allergic reactions to other vaccines, drugs, or food, they want you to talk to the health administrators. If there's a weakened immune system, or blood thinning medication, or a bleeding problem, then again, they want that discussion to happen. If you're pregnant, or you think you may be pregnant, or you're planning on becoming pregnant, or are breastfeeding, again, they want that conversation to happen. What the US is doing is that it's actually allowing women to opt in if they want to get the vaccine, and if they're pregnant or breastfeeding. So they're leaving that choice up to the woman in question, just because there are almost 1/3 healthcare workers in the US that are currently pregnant or breastfeeding approximately. And so they just want that decision to be in the hands of the women if they would like to take that. So the vaccine is 2 dosages. It's again administered in the arm intramuscularly. It's 21 days apart. So I don't know how that's going to work with people. Like, I don't know if you're going to get a call in 21 days and you have to be back to get a vaccine, or if they're going to be a little bit flexible with that schedule. So that's something that'll be, I guess, dependent on the vaccine schedule or the vaccine availability and who's around, I guess. There are still some things that we don't know. So with the Pfizer vaccine, we know that it seems to protect against COVID, but we don't know anything about asymptomatic transmission. So what I mean by that is that you can get the virus and you can still shed it even if you're protected from disease. So you might get it and then pass it on to someone who might be not vaccinated, for example. So that kind of asymptomatic transmission, we have no information on. We don't know anything about long-term immunity. So how long does that immune response actually last? Does it last only half a year, a full year, or longer? So that's going to have implications for if this is going to be an annual thing or, you know, every 4 or 5 years. We don't really know. And safety, like, we know that it seems quite safe, and there's no indications that that'll change. But they are going to follow it up long term to make sure that there are no really rare safe side effects that are like 1 in a million or something that pop up. So again, that rare side effects thing. And then again, that vaccine safety in certain populations. So pregnant women, children, immunocompromised individuals. It's not that it's not safe in these populations. It's just that it's not been tested in those populations. Pfizer, so there are certain clinical, so some data that people do with understanding whether the vaccine is safe in pregnant women or not. So you vaccinate pregnant rabbits and then you see if that affects their babies. And essentially, Pfizer has said that they will be releasing some of that data for those experiments with their vaccine by the end of this year. So perhaps we'll get some information on whether the vaccine is safe for pregnant women soon. And so one question I get a lot, and I think this is my last slide, is that vaccine development was so quick, you know, were safety corners cut? And the answer is no. But everyone's sort of like I understand that, like, you know, it happens so quickly. Vaccine development never happens this quickly. And again, the answer is no, because that safety is the primary concern, and then it's the efficacy. So safety was the most important thing that everyone looked at this vaccine. The reason why the vaccine developed so quickly was because of this like intense international cooperation that happened. Everybody's working on it. You know, like when I've gone to a conference in my field and it has the world's experts in my field in one room, it's a pretty small room. Like there's not a lot of people that are studying what I'm specifically studying. And so, and another friend was describing, he's also in the doing research in Foothills, and he's like, in my field, you know, people have been researching this pathogen, this parasite that he studies for 50, 60 years. And when people are studying and doing research and discovering something, you write it up and then you publish it. And if you look at the number of papers is over the lifespan of that parasite being studied is so small in comparison to the number of papers that have been published about SARS-CoV-2 and COVID this year. And so everybody is working on it, and everybody is sharing that information with each other, and it's getting out very, very quickly. And there's also unlimited resources. Like usually when you're doing stuff in the lab, you apply for a grant and then you get rejected usually, and then you apply for another grant, and then you design your experiment and you get the data, and then you put it together in another publication, then you apply for another grant. So that is usually how it moves. There's a lot of bureaucracy, there's a lot of red tape, a lot of waiting around. Whereas in this case, all of that was cut. Like you needed money, it was there. You needed infrastructure, it was there. And so that is essentially the reason why we've gotten as far as we have, as well as because we're building on that foundation of looking at mRNA vaccine and research that has been done for 10, 20 years already that Nick was talking about. So Operation Warp Speed, for example, which is horribly named because nobody wants a very quick Tylenol or quickly approved Advil, but they put $10 billion USD to help with vaccine development. So Pfizer was mainly self-funded, but Moderna got almost $1 billion to work on their vaccine development. So when you have that level of finances in your disposal, you're able to get things done very quickly. And again, those clinical trials, Pfizer had almost 40,000, Moderna had 30,000. That number is amazing that many people sort of volunteered for these trials. So I think those are some of the reasons why we got this vaccine very quickly. And the fact that vaccines take so long in normal circumstances is actually not a good thing. It'd be much better to be able to retain some of this lack of red tape and bureaucracy going forward post-pandemic. So with that, I'm done talking. And so if there's any questions you have, I'm happy to answer.
Great, thanks guys. Some of the questions that some of our coworkers have submitted, I'll just run through those quickly. And in the meantime, if anybody has any questions they want to type into the chat, that would also be fine. A lot of questions we got were around pregnancy and fertility. So I think Rye pretty much covered mRNA vaccines being safe in pregnancy, post-pregnancy, pre-pregnancy. However, we're not included in the data groups. Maybe, Rye, do you want to address, there was a concern linking fertility and COVID mRNA vaccines. Do you want to talk about that quickly?
Yeah. And so again, like we don't have any evidence to say that it's not safe in pregnant women. Like a lot of people have sort of discussed this, that people with like gynecology backgrounds and immunology backgrounds, that this vaccine is likely quite safe in pregnant women. But again, until that data comes in where it's actually tested in these pregnant rabbits and whatnot, people are being cautious. But the sterility / fertility thing, there's been some, I guess, claims that I've came across where people are worried that the vaccine will make it so that you're sterile or not able to conceive. I believe that's a question, right? Like looking at that claim.
Yeah, those specific claims, yeah.
Yeah, and so that's kind of interesting because that sort of comes from the idea that the spike protein is similar to a protein that's found on the placenta. And if you actually look at those two proteins, that's not really the case. The protein is huge, and there's only a very, very, very small segment that's similar. So it's kind of like saying, the analogy I was using is that if you've got a cake and you build this like 3-tier cake, this is the spike protein, and you decorate it with like white whipped cream dollops on the top. The other protein, or the placenta protein, might be the same, like the only thing similar might be that one white dollop, not the entire cake, so to speak. So they're very, very different. But also rationally or logically speaking, if that were the case, then if you got COVID, you would actually become sterile and infertile. So that has not been something we've observed. People who have gotten COVID, women who've gotten COVID have been able to get pregnant. So that's not really a concern. I hope that answers the question well.
I love baking analogies. Yes. And then one more question. Are there specific concerns about mRNA vaccines and breastfeeding, or was this group not included because there was not enough data?
It's the latter. It's not actually, it was not included in that group. So oftentimes, just historically speaking, pregnant women who are breastfeeding are not often included in clinical trials, just because they want to be absolutely sure that there's nothing that can go wrong. So they're often included after like some of these initial clinical trials are already done. So in this case, it's just that they weren't included in the group. But again, if you talk with people that sort of have the background in immunology and vaccine development, etc., they'll say that the chances of that mRNA going, from your arm to your breast milk and then into the infant or the baby's mouth, by that time that happens, that mRNA is just like long gone, because it again degrades in that like 10-hour time period. So it really shouldn't be a concern, but again, it's just because that the group wasn't included in the clinical trials.
Okay, great. Next section we have is viruses mutating. So next, some of these were for you. If the virus mutates, are we going to need a different vaccine, and is the virus mutating now?
So the virus is mutating now. The problem is we don't know what these mutations are doing. Like, to be able to know for sure what these mutations do, you'd have to do a head-to-head comparison with a virus that has the mutation versus a virus that hasn't. And those experiments haven't really been done, just because you need access to a facility that allows you to do experiments with SARS-CoV-2. So we can speculate as to whether these mutations can increase infectivity, increase transmissibility, make it better able to spread, which is a good assumption, because if something is highly prevalent, if a certain mutation is suddenly highly prevalent in a population, and it's a good assumption is that it's making it to some extent easier to spread, but we don't know that for sure. And the virus, in terms of evading the vaccine, there have been some spike mutations. There have been several spike mutations that have been identified throughout the pandemic. But again, we don't know what impact that will have on the ability of the virus to evade the vaccine. So if it's, and again, it depends on the extent of mutation, because the spike protein is a large protein, and to change it in a substantial way such that the virus will be able to evade immune response, a few different mutations will likely have to happen. So to sort of tie a bow on this answer, yes, we've seen mutations. For the most part, we don't know what they do. For the most part, the news reports that have been sort of alarmist about the fact that this virus is mutating, it doesn't necessarily mean anything, because viruses mutate all the time. They replicate so fast that it's just the nature of how they propagate and spread. And there was concern that the spike, that sort of all of the vaccines that are in development are focused on the spike protein, just because of the possibility that with a few mutations, all of the vaccines could fail. But for the most part, there hasn't been enough mutations, and combine that with the clinical trial data that has showed that these vaccines targeting the spike protein can protect against disease, and the viruses are not evading the immune response. All of that taken together suggests that for now, there's not really much to worry about in terms of immune evasion from what we've seen so far. But yes, that immune evasion is always a risk, especially when all of our vaccines are targeting the same molecule. Sorry,
okay,
that was long-winded, but hope that answered that question.
I find that less comforting than the answer I was hoping for, but somewhat comforting.
Well, I mean, I just want to quickly add a little bit to that. The spike protein is also constrained in how it can mutate, because that spike protein still needs to bind to that ACE receptor and enter. So it can't just like change willy-nilly. Like if it changes so much that it doesn't bind to the ACE receptor, then our problem is gone. It can't affect us and infect us anymore. So there is a large part of that protein that does need to stay very similar or to what it is right now, because it does need to be able to bind to our cell receptor and then enter. And so as a result, that might actually limit some of that mutation possibility. Where if you
Okay.
have a mutation there, it's not going to be helpful, because again, that virus, all it wants to do, like Nick was saying, that the virus's best life is just like getting into our cells, replicating, and then going into a new cell and replicating. And so if it can't do that, it's going to die off.
Oh, okay. Interesting. Next question for Nick. If you have COVID symptoms after you receive the vaccine, does this mean you're contagious?
No. So with the mRNA vaccine specifically, again, you're receiving only a part of the virus. So it's the RNA molecule that encodes the genetic information, the genetic instructions that's required to make the spike protein, and it's not the full virus. So you can't get COVID-19 from the vaccine. It's impossible. So no, you wouldn't. The symptoms you would be feeling after getting one of these mRNA vaccines would be not due to actual infection by SARS-CoV-2, because it's impossible without the full virus.
Okay, great. The next section of questions we had was with administration and cold chain. One of the questions I got a couple of was, why was the vaccine ready so fast? But I think Rai covered that. Another question we got is, will this vaccine be a yearly thing or a couple doses? As Rai mentioned, I think the scientists don't know yet. And then maybe one you could answer is, why does it have to be stored at -80 and -20 for the different vaccines, and the implications of administration because of this? You're on mute.
Sorry, I just realized that. Yeah, so the Pfizer vaccine is -70 storage, and the Moderna vaccine is -20 storage. And so I'm not entirely sure why there's different temperatures. They're both very similar. They're both mRNA for the spike protein in a fatty bubble. So maybe the fat composition is a bit different in the vaccine, the two vaccines. So maybe that's why you've got different temperatures. So the Pfizer vaccine, because it's -70 or 60 to 80, it means that it's just not going to be feasible to get it to most places, because the -70, -80 freezers are not readily available. And so you might have more places that are like hospitals or certain sites where you can store them. It's not just about storing them, it's about transporting them. Like, that's really the issue. Like, transporting something at -70 is challenging. -20 is a lot easier. So that might be why Moderna is going to be likely used in some of the more remote areas up north. So Health Canada has not approved Moderna yet. However, they've already purchased a whole bunch of -20 freezers, and they have already delivered said -20 freezers up north, and they're already doing dry runs, practicing getting stuff that could be the vaccine up there in those conditions, so that when it gets approved, it'll be delivered readily, and just you can hit the ground running essentially. So there will be some implications on administration in the cold chain, and so the logistical challenge is quite massive for making sure everybody gets vaccinated. The AstraZeneca vaccine is a little bit interesting because that one, it doesn't actually require that much of a cold chain. I believe it might be room temperature or fridge. I have to recheck that. And so that is also very, very good, because it might be the vaccine that a lot of other people get, especially in sort of the developing area where it's even harder to have that cold chain. So the fact that we have different vaccines at different abilities to be administered in different ways is a good thing.
Great, thanks. We had a question about what resources could we direct people to look into, or if people were asking for more information about this, but maybe in the interest of time and accessibility, we could email those resources to anyone who's interested. Two more questions here. We had one coworker who's very concerned about when he can go back to attending concerts. Nick, could you provide your feedback?
That's a good question. I'm wondering that myself. I suspect that most of the population, at least here in Canada will be immunized by fall, or immunized and have immunity by around this time next year. So depending on the uptake of the vaccine, like if absolutely everybody takes it to the point where we have herd immunity, then by early 2022 we might be able to go back to huge crowds and at concerts and things like that.
Okay, great. That person's not on the call, but I'll let him know. The last question here in the chat is there any specific allergies that make people more susceptible to having an allergic reaction? I don't know if either of you want to cover that.
So I don't think that's known, or I haven't read anything to that effect. Essentially what I've seen is that there were some allergic reactions that people in the UK were, like, I think it was 2 vaccine recipients who had allergic reactions, and they had essentially a history of having severe allergies. So they had their adrenaline auto injectors or something of the sort with them, and so they just used that and then they were fine. But essentially Health Canada lists that, has a list of the ingredients, and if you know that you're allergic to any of those ingredients and or you have allergies to foods or drugs or vaccines from before that you're aware of, they're asking you to bring that up and have that conversation with the healthcare provider. I don't know who that is, because I mean, you guys are the healthcare providers, so I don't know who you talk to, but yeah.
Okay, great.
Ask the question in the mirror.
Okay, I watched a webinar from Health Canada a couple days ago where they said anaphylaxis okay. Polyethylene glycol was a contraindication. Thank you, Sean. If anybody has any more questions, we could cover those, but otherwise, thank you very much for attending. I hope you got something out of it. Yeah, I guess that's all I really have to say.
Yeah, we can stick around if other people have any questions, or if people need to go, that's cool.
In terms of resources, I would highly recommend the podcast This Week in Virology. It's sort of above the level of understanding for the average layperson, but I mean, my parents like to listen to it, and at least my dad is a non-medical, non-scientist. So that's something you could look into. If
you talk to Jarna, she's an avid TWIV listener now. She'll tell you all about it.
It is a bit jargony though, but it is really nice. And they can be quite long, but they're great if you just plug them in and then, I don't know, make dinner.
Yeah, okay. Similar to how the first 10 minutes are really awkward on Zoom calls, I think the last 10 minutes are really awkward as well. So we will shut this down, and I'll see everybody at work next time we're there.
Thank you both so much.
Yeah, no worries. Yes. Thanks for attending.
Yeah, thanks for listening.
Yeah, that was really great, guys. And thanks, Noelle, for organizing.
Yeah, no worries.